All right, can everyone see my screen? Okay, well, thank you for coming. It's a pleasure to be here with you all. I'm Dr. Marcy Kingsbury. I'm an assistant professor here at Harvard Med and the Lurie Center for Autism, and I do translational research using animal models as well as working with clinicians to do some human intestinal organoid work. So today we will be talking about gut health, the microbiome, and the GEMMA study, and I will also briefly mention a gut-brain axis study as well. So the outline for tonight's talk, Sarah and I will be trading off. I'm going to introduce the gut microbiome as well as the gut-brain axis and the microbiome gut-brain axis. And briefly introduce some animal models we use to study the communication between the gut and the brain. Then Sarah will come in and talk about some clinical background regarding the microbiome and GI symptoms in ASD patients. And then we'll introduce the translational work that we're both involved with. I'm just going to move my bars a little bit so I can see a little bit better. So as we all know, autism spectrum disorder has core symptoms that include social behavior deficits, impaired language and social communication, as well as restrictive repetitive behavior, increased anxiety, as well as sensory deficits and sensitivities. It's increasingly recognized as a whole body disorder, given the variety of gastrointestinal symptoms, dietary issues, and immune dysfunctions that have been reported in individuals with ASD. And often the core symptoms of ASD are examined independent of these comorbidities that may in fact contribute to some of the behavioral hallmarks of the disorder. And so some of these comorbid symptoms include GI symptoms, the dietary feeding issues, the immune dysfunctions, and these GI symptoms can include constipation, diarrhea, gastric reflux, abdominal pain, and visceral hypersensitivity. And these GI symptoms have been associated with irritability, social withdrawal, stereotypy, hyperactivity, as well as language regression. And some studies have shown that the GI symptoms can correlate positively with the severity of core behavioral deficits. So now I'd like to turn to a closer look at the gastrointestinal system, and particularly the bacterial and species that live within the gut. And I want to introduce the microbiome. So in order to introduce the microbiome, I'm going to first define microbes, and these are extremely small living organisms such as bacteria, fungi, viruses, and parasites. And these are just two examples here shown in these pictures of colonies of these small living organisms. So you can see colonies of fungi as mold on bread, and colonies of bacteria if you were to look at agar plates grown in the lab. So the microbiome just refers to the collection of these microorganisms that are living within a particular environment. And when we talk about the human microbiome, it's the collection of these organisms living on the body at various sites. And it's quite remarkable because we have 3 trillion bacteria living on each one of us, and there's approximately 1 bacterium for every single cell in our body. And so it's important to know this ecosystem and to try to understand it. So the gut microbiome is the collection of these organisms that live specifically within the gastrointestinal tract. And they're important because many of these bacteria are commensal and beneficial, and they serve to train the immune system during development to differentiate between pathogenic versus non-pathogenic organisms, as well as to develop appropriate immune responses. However, if the pathogenic bacteria take hold in the gut, or if the gut ecosystem becomes unbalanced, then gastrointestinal symptoms can develop, as well as GI dysfunction. And so it's becoming increasingly recognized that individuals with not only neurodevelopmental, but neuropsychiatric disorders are not only characterized by emotional and behavioral deficits, but also by gastrointestinal disease and disturbances and alterations of their gut bacteria. And so shown here on the left side is healthy status, which is believed to be linked to healthy gut bacteria, healthy levels of inflammatory cells within the gut, and that translates into normal behavior and emotion. Where the flip side is that if you have unhealthy gut bacteria, you can have increased gut inflammation that can lead to abnormal behavior or abnormal emotion or cognitive processes. And for the past 5 years, I've been really interested in what guides one to a healthy versus a disease state, and whether abnormal CNS or central nervous system functions and abnormal gut functions are linked. And if they're linked, how might they communicate? And so that brings me to the gut-brain axis. And this just refers to the biochemical and neural signaling that occurs between the gastrointestinal tract and the nervous system. And when we talk about the microbiome gut-brain axis, this accounts for the role that the gut microorganisms play in this communication between the GI tract and the brain. And it's important to note that this communication via the gut-brain axis is biodirectional. And what this means is that neural processes can influence gastrointestinal function, and then gut processes can affect neural functions. And 2 examples of this is that when my daughter has a week of standardized testing, she will often report stomach aches during that week of testing. So I interpret this as her neural function, that being stress is impacting her GI tract and the pain that she feels. The other way that you can look at it is when you look at individuals with functional bowel disorders, such as Crohn's disease or ulcerative colitis, inflammatory bowel diseases such as those, they're actually at increased risk of developing anxiety, depression, sleep disorder, chronic pain, and memory problems. And it's believed that with their flare-ups of their gastrointestinal inflammation, they have flare-ups in these neural processes, anxiety and depression and the like. I'm going to briefly, before I turn it over to Sarah, just talk a little bit about the methods that we use to study gut-brain connection in animals. So one of the ways that you can study this connection is using germ-free mice. So these are mice that are raised in sterile environments that are devoid of all microorganisms. So these mice do not have any microbiomes, and it allows you to selectively introduce particular bacterial species into the gastrointestinal systems of these mice to look at readouts or outputs of behavior. Although it's not as applicable to humans because us as humans are not raised in sterile germ-free environments. Another method that's being actively used in studies is called fecal transplantation studies. And that's where you take fecal matter from humans or mice and you transplant it into a donor mouse. And this could be humans or mice with a particular disease state or altered gut microbiome. So you can see how that affects a donor mouse. Or you could have a mouse with a particular disease and you could transplant neurotypical human fecal matter or mouse matter to see if you could correct any of the physiological processes in the animal or perhaps behavioral deficits. Other common techniques that are used to study this gut-brain connection are the use of antibiotics, which will effectively significantly decrease the load of gut bacteria, both good and bad in the gut, to look what happens when you knock down this ecosystem and how that might affect behavior. And the probiotic studies are the selective addition of particular bacterial species, usually ones that we consider beneficial, to see how addition of these species to a gut ecosystem might alter or impact behavioral processes. And typically in mice, this is done through the addition of particular bacterial species or probiotics to drinking water and antibiotic treatments to drinking water, and then analyzing the animals if we're looking at social behavior and a bunch of social behavior tests. So now I'm going to turn it over to Sarah.
All right. So I am a pediatric gastroenterologist or GIA doctor at Mass General in the Lurie Center. And so we're hoping that you know, to kind of give you an overview of some of the things I see every day, and I talk about with you and your families, and hopefully give you some ideas about where we might be headed in the future. So there are many things that affect the microbiome. Some of them are things that we can do something about. Some of them are things we have no control over, like your age and your genetics and your gender. Diet affects microbiome. So what you're eating, what you're not eating. Exercise can affect it. Nutrition, so not just what you're eating, but sort of overall nutritional status. Infections can shift the microbiome, and inflammation can shift it as well. And so as I was putting this talk together, I think a lot of the slides say similar things, but just each in a slightly different way. And so as we're sort of reading through these topics, you can think that there are a lot of these that have some differences in children who have autism, like in terms of the diet or physical activity level, or being underweight or overweight or at an ideal body weight. The idea that, you know, infections or getting treated with antibiotics more often because you're uncomfortable and then your doctors can't figure out because you have trouble telling them what's going on. So for all of these reasons, I think that there might be sort of bigger shifts in microbiome within the autism population. All right, next slide, please. Sorry, we didn't get this coordinated fully, so we're going to do the two people's slide switch over. So as Marcy was saying, the role of the gut microbiota in health and disease. So down the left-hand side, just as a carryover from the slide before what we were talking about, so there are a lot of disease conditions, medical conditions that are associated with a change in the microbiome. So obesity has been found to have different microbiomes, diabetes, celiac disease, inflammatory bowel disease. People with lots of allergies have a different microbiome, asthma, cardiovascular disease, so like heart disease or high blood pressure. Some types of cancer are associated with the shifts in the microbiome, and of course, autism. So on the picture sort of shows sort of the progression. On the left side, the red things are things that are considered diseased state, and then on the right side is things that are considered beneficial or healthy things. And the lines across the top are sort of the inputs into things that can affect the microbiome. So the proton pump inhibitors, which are like medicines for acid reflux, antibiotics, those clearly have an effect on the microbiome. Different foods that you're eating, so whether it's saturated fat, sugar or fructose, or excessive protein consumption, can all affect microbiome as well. And those are sort of on the negative side of things. And then fiber intake and probiotics are considered positive benefits. And I'm not going to go into specifically what all the metabolic pathways are, but when we talk about later on the word metabolome, that's sort of referring to all of these different metabolic processes and all the chemical reactions that are taking place as part of the digestion process. And so there's a lot of chemicals and abbreviations in there, but the sort of coming down the left-hand side in the red, the SCFA is short chain fatty acid production. There's lipopolysaccharide production. There's increased cardiovascular disease risk. There's gut inflammation, insulin resistance, which is associated with diabetes, more of type 2 diabetes, with being overweight. And then as you get sort of towards the good side of things, the green side, with the beneficial inputs for the microbiome, that can increase your short chain fatty acid production, antioxidants, which protect you from what we call oxidative stress to the body, kidney toxins, improved lipid or fat metabolism, decreased gut inflammation, better insulin response, and a reduced risk of some infections. So everything is a spectrum in health and disease, and this hopefully sort of illustrates some of why it's such a complex pathway to understand. Next slide. This is a simplified way of understanding dysbiosis, which just means an altered biome or an altered microbiome in autism. So overall, most of the studies, I just picked out a few just as an illustration. Most of the studies in microbiome have been fairly small, but if you kind of look at them one after another, the overall themes are that in, I say kids with autism. I know some of you probably have adult children, but since I'm pediatric, I'm going to continue to say kids. But overall, in autism, the main findings in terms of the microbiome changes are that there's a decreased biodiversity overall, and that just means there's less variety. So, and then the other finding is that there are specific shifts in that the balance is sort of out of whack, and there are certain, too much of one kind or not enough of another. So the little picture on the left, picture A, is considered just a little graphic of a normal healthy intestine. You see lots of different colors, which represents, and lots of different shapes representing all the different species of bacteria and all the different components of the microbiome. And then on the right is just a graphic of dysbiosis, where things are not in good balance. So you can see just from looking at it quickly, there's not as many colors. So there's less of the variation, less variety, so less biodiversity. And specifically, there's an imbalance. Like I actually counted these when I found this, but there's more green on the right than on the left. And there are obviously like fewer of the other ones. So that's just the overall is decreased biodiversity, and and then specific things are out of balance. Next slide. And this again says similar things, but where that becomes important is that in the imbalance, one way of thinking about it is there's not enough of the good kind of bacteria and too much of the bad kind. And so you have to kind of get things back into a shift. This could be because of dietary selectivity or changes in the diet. And this can, like we talked about on the other slides, this can lead to the altered metabolome or all of those chemical metabolic processes that happen. One of the specific bacteria or category of bacteria that has been found in kids with autism is that there is decreased amount of lactose fermenter population. And so lactose is, as most of you may know, is the sugar or the carbohydrate component in milk and anything made from milk products. And so that may be why a lot of our kids have more trouble digesting milk, like a lactose intolerance issue is actually not just related to how much enzyme their body is making, but also the specific bacteria in the intestine that help to digest it when things are in a good balance. Next slide. So I'm going to go through just a couple of studies. This one is from 2018. Rose et al. Looked at children with gastrointestinal symptoms. They were 3 to 12 years old, and they found that the children with autism had a different gut microbial composition when they were compared with 41 typically developing control patients who also had gastrointestinal symptoms. So two groups both had GI symptoms, but the patients with autism had a different microbiome. Next slide. Here's another study. So Kang et al. Looked at a study of 40 kids also. Again, see, these are small numbers, 20 with autism and 20 without autism. Similar age group, 3 to 16 years old. And they found that the children with autism had lower levels of several specific species. So Coprococcus, Prevotella, and I'm not even going to try to say the V one, but they had decreased numbers of those. And so those are thought to be maybe the beneficial ones. So this is a category where they have not enough of the good kind. Next slide. There's a few more. So again, looking at, so there's been other ones looking at Clostridium, Ruminococcus, Cetterella. It's like a different language, but most of these are just different bacterial species, again, that are found to be different in children with autism as compared to children who don't have autism. Next slide. And then this is another one specifically looking at children with autism who also had GI symptoms. And again, some more vocabulary words, but also in the Clostridium species. This was from 2016. And they were looking at also at the neuroimmune signatures with functional abdominal pain in children with autism. Here's another one. So some of those other ones that I mentioned are considered are just sort of cross-sectional studies, meaning you just look at one snapshot in time. What are we dealing with right now? And those are a really good starting point. This one was actually a prospective study, so following kids over a longer time point. Over a longer period, so at multiple time points. This was looking at 69 neurotypical children followed forward in time. And they found that, again, pointing to maybe some beneficial bacteria, the kids who had a higher number of a Bacteroides species when they were 1 year old were found to have better expressive and receptive language skills when they were 2 years old. So this is interesting, right? So if you follow even earlier earlier in time and you follow over time, there might be some signals earlier on about some good balance in the microbiome. Next slide. Okay, this is another study looking at the infant microbiome and neurodevelopmental. The previous one was up to 2 years. This one is at 3 years. It was really meant as a vitamin D study, but they had sort of this other things that they looked at too, because they had so much data. And for this one, they found so it was a study of 309 infants with fecal or stool samples, and then assessing their development and preschool age at 3 years old. And they found a statistically significant association between the gut microbiome and infancy and several developmental measures. So fine motor, communication, personal and social skills at 3 years old. So this is supporting the hypothesis that the microbiota early on in life is associated later on with neurodevelopmental outcomes. Next slide. Okay, so hopefully, like I said, those were just a few studies that look at sort of establishing there are some differences in children with autism in the microbiome. The main question is, what can we do about it? How do we treat that dysbiosis? From the early slides where we talked about some of the factors that can influence microbiome, some of these should make sense. So maybe there are changes in the diet that can influence the microbiome. Antibiotics, as we discussed, prebiotics, probiotics, ultimately the fecal microbiota transplant, or FMT, which is basically the poop transplant. And it's sort of a chicken and the egg, right? So we know that there's an association with autism and having an altered microbiome, but we don't really know what came first. We don't know if one is causing the other. And like Marcy said, it most likely is going in both directions. Okay, so here, a couple of slides about probiotics. Just starting with a mouse model of autism. So there was a mouse model of autism who were treated with a specific probiotic called Bacteroides. And they found that giving this probiotic changed the gut microbiome profile. It changed the blood metabolite profile. So all of those chemical reactions that we talked about. And it corrected the increased gut permeability as well, and improved some of the social autism behaviors. Some more evidence that addressing the microbiome differences could be helpful. So there was one study that looked at a specific antibiotic called vancomycin, and they found what's different or specific about this one is that it's considered a non-absorbable antibiotic. So most antibiotics, you take them by mouth, it circulates throughout your bloodstream, and that's how it gets to wherever the infection is. But vancomycin is made specifically to stay in the intestine. And so it doesn't circulate, it only stays in the intestine. And so it makes sense that would go directly to where the bacteria are. So they found that treatment with vancomycin improved both GI symptoms and autism symptoms. However, those improvements were temporary and they only lasted a few weeks. And as some of you may know, based on your own experience in your own families, there has been some anecdotal response from families for other interventions, like dietary changes, like a gluten-free or casein or milk-free diet, sometimes even more restrictive than that, probiotics and antibiotics. But so far, we don't have enough big evidence to use those as recommendations yet. Ultimately, so I sort of think of it as the ultimate way to change the microbiome is with the FMT, the fecal microbiota transplant. Right now, it's only FDA approved for a specific and recurrent infection in the colon called C. diff. There are ongoing studies looking at FMT for a different condition called inflammatory bowel disease, so like Crohn's and ulcerative colitis. And then there was a recent pilot study in humans that did show improvement in GI symptoms and autism symptoms. So this was published out of Arizona in 2017 with Kang et al. It was a small trial, so only 18 children. They all had autism, and they did find there were some baseline microbiome differences, like we've been talking about. So the kids with autism had the lower levels of the good bacteria, and then the bifidobacteria and Prevotella. So, but as for their protocol, they did a 2 weeks of antibiotics. They did a bowel cleanse. So like some of you may have done with me, if for major constipation or prior to a colonoscopy, we'll do a full bowel clean out. So that was part of their protocol. And then they did 8 weeks of the FMT, so the actual stool samples, the stool transplants. Their initial protocol included vancomycin, that non-absorbable antibiotic that I mentioned before, omeprazole, which is a medication that an acid-reducing medication, and then the bowel prep. And then they had every day they had a standardized human gut microbiota treatment. I can't remember if it was in a capsule formulation or how they did it in this particular study. But they had very promising results. They found an 80% reduction in GI symptoms in these 18 children who were participating in the study. They repeated the microbiome testing. They found significant alterations in the microbiome. And they tested again 8 weeks later, and they found that the changes were still there, which is better than what's like previously some of the probiotics was like a short-term response, but they felt that the 8 weeks out was lasting longer than what they would have expected compared to the probiotics. They found that the FMT resulted in improved behavioral autism symptoms. They reported a 45% decrease in autism symptoms compared to baseline. They used the Vineland, which is a developmental test, and they found that just in this 8 weeks of FMT, the average developmental age increased by 1.4 years, which is kind of a staggering number. And they reported that 83% of the participants at the beginning were rated into as having severe autism, but at the end of the study, only 17% fit into the severe category. 30% were categorized as mild or moderate based on symptoms, and 44% didn't even meet the cutoff for mild autism symptoms at the end of the FMT. Not surprisingly, every family returns 2 years later to provide some follow-up data, and the family still reported ongoing benefits. So they reported a 58% reduction in GI symptoms compared to their baseline before they started it. And most parents reported slow but steady improvement in their core autism symptoms as well. There are some limitations. It was a very small study. There was no control group, so everybody knew that they were getting FMT, and there was no placebo group to compare it to. But it's very promising and very encouraging. And so this is where a lot of, this is sort of the foundation for some future research studies as well. So we need validation studies to see if these numbers can be replicated. We need larger numbers. And then eventually we'd like to establish the causality. So not just the association of autism and microbiome changes, but is there a way that that one is causing the other, so that we know where in the cycle to adjust some of our management strategies. So how can we keep the gut microbiome healthy? This is just a cute graphic I found online. I think again, targeting that screen we talked about at the very beginning, some of the different factors that can affect it. So trying to reduce stress, getting physical exercise and activity, avoid antibiotics if they're not necessary. Think about supplements like prebiotics and probiotics, eat a variety of different foods. Try to eat fermented foods, so kimchi, kefir, yogurt, sauerkraut are all fermented foods that have some probiotic benefit, and then trying to reduce artificial sweeteners as well. For now, as some of you may know, for now, there aren't any clear recommendations. So we are really hoping that maybe over the next few years, we'll have a different conversation around the microbiome topic. For now, I would say probiotics might help. They don't help everyone. And at least right now, we don't have a way to predict who will respond or which probiotic to use. So it is a bit of a trial and error. It's not going to be an immediate quick fix. It takes, I would say, 2 to 3 months to shift the microbiome over. So don't give up after a week or so if you don't see any significant differences. And just as a cautionary measure, the supplements are not regulated by the FDA. And so there can be some variation, from one batch to another about what you're getting with supplements. I would say, if taking medicines is a challenge, I don't know that this would be a battle that I would say is worth fighting. Like for a picky eater, I'd probably say take a multivitamin first over trying the probiotic as sort of a starting point. But down the line, if you're making progress, or, if medicines are easy to do and you can find one that your child would like, then I think it's worth a try because there's a chance that it could be helpful.
Okay, thank you, Sarah. I'm going to switch back and I'm going to talk about a translational study that I've been working on, and then Sarah would like to introduce and talk about the GEMMA study, and then we'd be happy to open it up to a conversation. So I'm just moving my menu a little bit. So we call this the GBA study, and Sarah is actively involved in helping us to recruit patients. And so what we're interested within this study is how gut microbes might contribute to the core ASD behavioral symptoms. And we were interested in this study because it's been noted, and this has been done in collaboration with Dr. Alessio Fasano, who's the director of the Mucosal Immunology and Biology Research Center here at MGH, and the chief of pediatric gastroenterology. And so what we've observed in the literature is that individuals with autism have been characterized by increased intestinal permeability, which is something that Sarah has just mentioned. And one mechanism for an increase in intestinal permeability is the presence of haptoglobin 2 allele, or HP2. And this allele is linked to zonulin production, so that individuals with either one HP2 allele or 2 can produce zonulin. And zonulin is a protein that is a known regulator that can regulate the tight junctions or the spaces between the cells that line the intestine. And so when zonulin production is stimulated by the presence of this allele, it opens up the spaces between the intestinal cells here, and it allows for bacteria and food particles and antigens to diffuse through the space and enter the bloodstream and cause inflammation. And so this is what we term a leaky gut. And interestingly, zonulin production is stimulated not only by gluten and is involved in celiac disease, But zonulin is also stimulated by gut bacteria, and particularly gut dysbiosis, or an imbalance of the gut ecosystem. So we thought that having an HP22 genotype as opposed to an HP11 genotype may predispose individuals to increased intestinal permeability. And when we looked at our patients that are being recruited into our study here at MGH, and we have at this point more male subjects than females, but these are males coming in for a clinically indicated biopsy. And we find an overrepresentation of the HP22 genotype as opposed to the HP11 shown here in the table. So these are patients that are involved in our study. And these are patients with GI symptoms. So it seems like those that have GI dysfunction, there's a greater prevalence of the HP2 allele. One thing that we wanted to address, or a question that we wanted to address, is it's currently unknown whether gut microbiota from ASD patients with GI symptoms exacerbates this intestinal permeability and blood-brain barrier permeability to contribute to core behavioral symptoms. So this is, again, what we wanted to address in this study. So our hypothesis was that an increased genetic predisposition for increased gut permeability, that being the presence of the HP22 genotype, may synergize with gut microbiota from ASD individuals that have GI symptoms to upregulate the production of zonulin that is able to be produced due to the presence of the HP2 gene. And then this upregulation of zonulin allows greater diffusion and opening of the tight junctions to allow greater diffusion into the bloodstream, increasing inflammation and potentially leading to behavioral deficits. So the way that we're addressing this with this study is that with Sarah's help, we're able to recruit patients that are undergoing duodenal biopsies. And we take these biopsies and we genotype them for HP11 or HP22. And we create intestinal organoids, which are clusters of intestinal cells. And then we plate them out into an intestinal monolayer. So this is an in vitro culture system that's approximate the intestinal epithelial barrier. And what we do then is we perform baseline assessments of gut permeability and inflammation. And then we come in and we expose our intestinal monolayer cultures to stool obtained from neurotypical healthy individuals without GI dysfunction, or ASD pooled stool from individuals that have autism that also have GI dysfunction. And we look at how this exposure to the supernatant from these pooled stools affects permeability, antigen trafficking through the intestinal monolayer and inflammatory processes. We also have an animal model in addition to using human intestinal biopsy tissue. And here we have a zonulin transgenic mouse that actually has the HP2 allele inserted into its genome. And so we're able to breed these mice for the HP22 genotype and zonulin production versus our wild type mice that do not produce zonulin. And we can do a similar assay as we were doing with our intestinal organoid cultures, where we gavage the mice. We're basically doing a fecal microbial transplant, where we gavage the mice with stool from either control individuals or those with ASD with GI symptoms. And we're really interested in whether the exposure to these different types of stools is exacerbating social behavior and anxiety-like behavior. And these are just some of the behavioral tests that we can do with mice to assess this behavior. So we can put our test mouse that has had the FMT into an arena to test sociability, whether it wants to go and engage and investigate a novel mouse, or the other chamber contains an inanimate object. So we look at how much it's interested in engaging with a social stimulus. We can also assess social novelty. We can expose the test animal to a novel mouse that it's never seen before, or its cage mate and see whether it prefers to hang with its familiar cage mate or, again, be more ambitious and engage with a novel social stimulus. And these are two tests that we use in the lab to study anxiety-like behavior. And as you might imagine, this might produce anxiety in a mouse, perhaps like climbing a mountain for us. So this is the elevated zero maze, and the mouse can choose to enter the open arms where it can hang off and dangle off the side and is not protected, versus the closed arms, which is presumably less anxiety provoking because it has more security. And similarly, in our light-dark assay, mice that enter the open light area of the arena are presumably a little less protected and are engaging in a little bit more bold behavior than in the covered dark compartment. So those are the two arms of our study where we're actively investigating the synergy between this HP22 genotype that is able to produce zonulin, that is a key regulator of gut permeability, and how it might synergize with the gut microbiota of patients, ASD patients that have GI symptoms. And one exciting thing about this study is that we actually have an inhibitor of zonulin that we can then follow up in these studies. And this inhibitor can actually block zonulin signaling, so effectively put a brake on that intestinal barrier permeability, and we can see if that could potentially be used as a treatment to ameliorate the permeability issues, even in the presence of an altered gut microbiome. So I'm going to turn it back over to Sarah now, so she can introduce GEMMA.
Thank you. So GEMMA is my big study that I'm working on now, and it is it's a longitudinal observational study. Then we're looking to see if we can identify potential biomarkers in young babies and children that are predictive of autism development. So the study enrolls infants that are 0 to 6 months old who have a sibling that's already been diagnosed with autism. We're collecting biospecimens, so blood, stool, urine, and saliva from the babies every 6 months until they're age 3. And then we are also doing developmental assessments, again, every 6 months, starting at 12 months of age. So looking at the biomarkers and then following with developmental assessments along the way. Next slide. Let's see. What's really cool is it's an international study. So as Marcy mentioned, Dr. Fasano, my chairman in GI, is from Italy originally. And so he's been able to sort of spearhead this, this collaboration. So we at Mass General are one of 3 clinical sites recruiting families and patients. So we're recruiting from patients from across the whole United States. All of the samples, the developmental assessments, everything is done remotely and through virtually. Italy is another site, and then Ireland is the 3rd clinical site. And then the samples are getting eventually distributed across, I think, 5 or 6 more countries across the world. So the US, Italy, the Netherlands, Ireland, Australia. Basically, we have all of these different specialists, experts in different parts of the metabolome, like the chemistry and the environment and epidemiology, who are going to look at all of the different samples. And what we're trying to see, to follow especially, is in the babies who do end up developing autism. Is there a point in time where we can detect sort of a biological signal or a biomarker that gives us a sense that that the metabolic pathways are changing, and then we're following the microbiome as well. So the autism assessments is one of the developmental assessments we're using is called the BOSA, which is the Brief Observation of Symptoms of Autism. And that looks at the core behavioral symptoms of communication, social interaction, restrictive and repetitive behaviors. And then we're also doing the Vineland Adaptive Behavior Scale, the developmental profile 3 and 4, depending on which language you speak, and the SRS-2 scales that are used together with the BOSA to get more information about the child's functioning in terms of physical skills, adaptive behavior, social, emotional, and cognitive skills, as well as communication. So these are very preliminary results, unofficial, but we just wanted to give you sort of a general sense of where we are now. So currently, of the babies who have been evaluated with the developmental assessments, currently about 33% or a third of them are showing some autism symptoms as measured by the BOSA, the developmental assessment. And we've hopefully been able to help with an earlier diagnosis than they would have gotten without being part of the study. As part of this, again, it's sort of a international multi-step project, and we're just one of 6 to 8 components of it. And so part of it, before we even got to the point of recruiting families, was that to get some preclinical data, looking at the role of the gut microbiome in autism and how it interacts. So I didn't do the actual studies. I'm not a mouse. I'm not in the lab with the mice, but this was similar to some of the things that Marcy had mentioned earlier. So I just wanted to share this with you. So they transplanted the germ-free mice with stool from children who had been diagnosed with autism with or without gastrointestinal symptoms, and then also different mice got transplanted with stool from their siblings as the control group. And then the samples were collected and pooled for transplantation into mice. And then let's see, next slide. So in the germ-free mice, treatment with the autism and GI stools showed decreased exploratory behavior. So some of those anxiety tests that Marcy had shown with the mice. In the germ-free mice who got an FMT with stools from the kids who have autism, either autism without GI symptoms or autism with GI symptoms, transplanting those stools into the germ-free mice did not induce a difference in behavior or neuroinflammation compared to the sibling control transplanted mice. And so they were looking at this sort of highlights some alterations in the microbiome and physiology, and hopefully provide some more evidence and some scientific background to thinking more about using FMT as a tool to study the autism physiology and potential effects of treatments. Let's see, and that is, I think, all we have for now. So we do have a few minutes for questions. I've been trying to preview them, and I think my initial comments are these are really good questions, and I don't know that we have answers for them, but we can take a look and see what we can find. Let's see. I don't know, Anne, do you want to call them out, or we can try to look through. Let's see. I'll see if I have answers to any of them. Maybe I'll start with those. Let's see, someone asked about, has anyone looked at autism in populations that eat lots of fermented foods, such as kimchi in Korea? That's a really good question. I think, I don't know the specific answer. I know there are definitely some of the studies that showed up that were from Korea. So it would be really interesting to compare sort of different geographic and cultural microbiome data. I don't know, Marcy, do you know anything specifically about microbiome in different cultures?
I don't.
Let's see. I'm trying to see if I can see any that have an answer, a direct answer. Has anyone done a study that looks at the correlation between plant diversity in the diet and frequency of autism? I've been reading about the gut microbiota for my own health and have read that greater plant diversity in diet leads to better gut microbiota health. So not just diet diversity, but specifically plant diversity, I guess is the question. I can't speak to like diversity in plants specifically. There was a whole study that I actually didn't have time to put in as a slide that sort of went through all the different food components and what the different microbiome changes were. I'm trying to think, there was cheese, there was other dairy, there was, but I don't know specifically about plant diversity.
I would say that I haven't seen a study specifically about plant diversity. I think it's a great question, but what I do know is that the bacteria in the gut, they like to consume fiber. And so if you're eating high rich fiber foods in the form of fruits and vegetables, then you're actually feeding the bacteria that can ferment and create those short chain fatty acids that Sarah had shown are really important for gut health. And I think in general, diversity is better. So a diversity of foods might allow more food for the different bacteria in there to generate diversity of metabolites. So I think it's a great question, and I think it would make sense because we do know that fiber is the food for these bacteria and are able to feed them.
So it looks like there's a couple questions for Dr. Kadzielski. So is there a specific probiotic you would recommend trying?
No, there isn't. I didn't actually say, so for GEMMA, part of that, the sort of the next step that we're working on also is an intervention step. So for the babies who do develop autism, we're going to offer a probiotic as an option for those families. And we went through a pretty extensive review of the literature, and we picked out some, but just like all those studies I was saying that showed different balances of probiotic or different balances of bacteria in the microbiome changes, there wasn't an overwhelming probiotic or just specific strain to use. In terms of what's commercially available too, it's probably going to depend more on what you can get your child to eat. So whether, if it comes in a liquid or a capsule or a powder. And I mean, some of the ones that have shown up, so like Saccharomyces has shown up, Bifidobacterium species, Lactobacillus species, but there isn't one specific one that I would suggest. Unfortunately, hopefully in the next couple of years, we'll have a better answer to that question.
And what is your opinion on limiting sucrose or fructose in the diet?
That's a big question. I think in general, I would say try, do your best to offer a wide variety of foods. And if your child is a picky eater, try to get the support you can for feeding and sort of gradually branching out. In terms of specific elimination diets, again, I think everything in moderation. And then if you are noticing that there's specific foods that tend to be triggers for GI symptoms or behaviors, then be more selective. But it's hard because there isn't. I think there's probably different subtypes of kids with autism or people with autism that respond to one thing, and some people respond to another. And it's not everyone's going to respond to a gluten-free diet, but there are definitely some families who found it really helpful. So I think just trying to get as broad of a diet as you can.
We maybe have time for 2 more questions. How about what is the relationship between microbiome differences in ASD and functional disorders?
Let's see.
And someone, feel free to unmute if you want to clarify the question too, whoever asked.
So there are microbiome differences in autism, and there are definitely microbiome differences in functional disorders. I don't know if the question is like the autism versus functional. Oh yeah.
I can speak to it. Hi. So if the kid has exhibited all these terrible GI symptoms for years, and they were diagnosed as having, like, say, a motility issue, mm-hmm. Is that a totally separate thing than the microbiome question in autism, I guess, is my question.
It's a really good question. You guys all have great questions with no great answers. I think the answer could be yes or no. I think there are some kids who respond to treatment like for constipation and things get better. But if you're but there are some kids with constipation that do respond to like a probiotic or ways to manipulate the microbiome. So there's probably some overlap, although again, there might be subtypes within those two different groups. Does that sort of answer your question?
Yeah, thank you.
I guess this is the last one. It's the last one that came in, and so we'll stop there. Do you think there could be a link between the reduced microbiome of children with ASD and their mothers receiving IV antibiotics while in labor?
Yes. Go ahead, Marcy.
I would say this is potentially, it could be a link. That's actually a link that I wanted to try to investigate in a mouse model. I have talked with OBGYNs that say that administration is usually very short duration and very targeted. And so the impression was that because it was so acute, it might not have that much of an effect, but I think it's worth something investigating. What I will say is that we do know that the baby's microbiome is very sensitive to antibiotic treatment in the early postnatal period, but it will be receiving a lot of good microbial species through breast milk as well that can then come in and kind of further bolster it. So, but that's a great question and one that I've actually wanted to answer for some time.
Well, we are out of time, so thank you everyone for joining us.